Check. Good evening, everyone. Welcome to the second class of our People's University Marine Biology. And the world of fishes is so large we had to do two classes. So our second class, as you can see, is called The Wonderful World of Fishes. I just want to mention that Tuesday, if you're interested in lunch with books, there's a program with two authors of a book called Heirlooms, which tells the story of forgotten 8 generations of American frontier women from colonial Jamestown to Belmont County, Ohio. And these two people, Dr. Pamela Manners and Dr. Richard Bernard, both went to Bethany, and Dr. Bernard was an executive at Bethany. So that's Tuesday at noon. Our instructor this evening, some of you met her last week, Dr. Jennifer Ripley Stuckel, earned an MA in Marine Biology from the Boston University Marine Program, located in Woods Hole, Massachusetts, and a PhD in Biology from West Virginia University. Her research primarily focuses on the reproductive physiology of fishes and the influence of environmental factors. And her research has taken her down the coast of Baja California to study barnacle recruitment in the eelgrass beds of a bay I can't pronounce, to study brood pouch, I'm having trouble with my eyes tonight, function, and seahorses. You're going to hear about seahorses tonight, I believe. And so here she is, Dr. Stuckel.
Thank you, guys. Thank you for coming along with me for this week as well. So this week, since I introduced you to fishes last week, this week I'm going to share some of the research I've done over my 20 years of research experience. Because most people, when they think of fish researchers, they think we just go out snorkeling and count fish and watch them swim. But I am what's called an ethologist. And ethologists study the behavior of non-human animals. So fish fall into a non-human animal. And I happen to study everything from reproduction to parental care to communication. And so I'm going to show you some of these things today. Today we'll have a multimedia presentation. I'm hoping it all works. I've got some videos and some sound clips. And we're going to start with sound clips. So fish actually produce sound. And fish use sound for deterring predators. Fish use sound for calling on other fish. And so I'm hoping you guys can hear these. So we'll look at a couple different fishes and some of the sounds they produce. So let's see if we can get these started.
Directly above the tile. Type 2 male hovers above the nest. Type 1 male also had a mouth display. While not clearly visible, there was also a female in the back of the nest blocked by the type 1 male. The second video shows a type 1 male inside his nest with his head facing forward. Right above him is a sneaker male with his tail to the left. A female is directly behind the sneaker male. You can see her tail to the right that becomes more visible as the sneaker male starts to move around. The female is in an egg-laying position, one that is mimicked by the sneaker male. The type 1 male takes a nip at the type 2 male that darts out of the nest, followed by a series of grunts produced by the type 1 male. You also saw another type 1 male swimming across the top of the nest behind the hinds.
So those fish there, and I'm hoping you guys could hear them. Everybody could hear it, right? If you guys couldn't hear things, I'm going to stop that. If you couldn't hear it, I was hoping you guys would tell me about not being able to hear it. And so those are called toadfishes. And toadfishes are well known for producing sound. in all kinds of different capacities. There we saw males defending each predatory defense, trying to defend a female, and they also use it for females to attract, for males to attract females as well. I'm trying to do two things at once, so I can't actually talk and push buttons at the same time. Now I'm going to show you another one. This is called a croaker, and this is a guy who's just holding a croaker, and he will make the croaker make some sounds for us as well. So that's a croaker out of water, but they make sounds in the water exactly the same. Here's another guy who caught another fish, and he has him out of the water, but you can hear a slightly different kind of grinding, croaking sound.
So okay, it's a nice little gronko.
And so there's all kinds of sounds that fish produce. Usually they produce these sounds by grinding together what we call pharyngeal teeth, or teeth in the back of their throats. And you can kind of hear that hard kind of grinding sound with them. Now, when I did my master's research at Boston University Marine Program in Woods Hole, I actually did research on species-specific sounds. So in Africa, there are these lakes that hold fish species that are only found in that lake. So I studied Lake Malawi. And in these lakes, specifically in Lake Malawi, there's over 300 species of cichlid fish. And you have seen cichlid fish if you go to Petco and walk up and down the aquarium aisle. They have tons of these African Rift Lake cichlids. They're gorgeous colors. They usually have very aggressive displays where they flare out their fins, and they produce sound. Now, unfortunately, I can't share the sounds they produce because the data is owned by the laboratory that I worked in. But what I was able to do was determine that the sounds of one species are distinctly different than another species, and that's how they maintain the species. Because there are over 300 species, like I said, in these lakes. So how do you know who's who? How do you know you're supposed to mate with somebody? And they use the sound. They use the differences in sound production to determine if that sound is of my species or not. And I actually, we wanted to know if fish could hear these differences. Could they detect it? Now, we have a method in humans. So when babies can't tell us if they can hear or not, we always do an auditory screening on a baby before they leave the hospital. And it's called an auditory brainstem response. So we put these electrodes on the baby's head, and we can determine if their ears, if their auditory part of their brain is picking up the sound or not. So in fish, we did the exact same thing, but we made an auditory brainstem response chamber for fish. So here's a fish. He's being held in a tank with his head out of the water with some electrodes, just like your baby in the hospital. And what we did was we made an airproof or a soundproof chamber with an air table. So we wanted to make sure that the fish was detecting the sound waves and not the vibration. If you remember last week, we talked about fish. Fish have that lateral line, which detects vibrations in the water. We wanted to make sure they're actually using their ears, their auditory. And so we had to make sure that it was on an air table so it wouldn't be a bouncing effect. And by playing a sound in the room and having these electrodes on the head, we could get an auditory brainstem response and determine that these fish were detecting the exact sounds and frequencies that they were producing. So they can hear what they produce. This over here to the right is showing you a fish in their ears. Their ears are much like our ears. They are vertebrates. They have semicircular canals. They have bones in their ears. I'll show you some of the bones in just a second. The interesting thing about fish is they also have that organ called the swim bladder that we looked at last week. And that swim bladder fills up with air and allows the fish to swim up or down in the water column. So he can position himself at the surface or move himself down low. And so we have seen different species use a different amount of their ear versus the swim bladder, or connecting the two so they can really amplify the sound that goes to the ear so they can hear even more precisely. So up here you see these are fishes that just use particle motion. So they mostly use their swim bladder and its vibrations. Okay. And you can see then we see some that have a swim bladder, but it's really far away from the ear. So again, it's mostly just using particle motion. But as we move down here, we can see that the swim bladder touches or connects or has a very particular like mechanical connection between the two so that it can determine very quiet, very precise, very distinct sounds, and even which direction the sounds are coming from. So it's really cool how they've evolved depending on their function, what they need it for. And so here again is the ear. This is the ear of just a typical old fish. And they have what are called otoliths. Otoliths are ear bones. And otoliths form these rings with each year of growth. So it's much like the rings of a tree. We can age how old a fish is by how many rings are on their ear bones. And we do that quite often when we catch fish. We'll age them off their otoliths. Of course, that's a sacrificial age. But we've even seen with the chondrichthyes, remember the skates and rays, all the fishes that had the cartilaginous skeleton instead of the bony skeleton. They all have the same type of ear structures that we have, the 3 semicircular canals, but they have modified them again depending on their body shape and their way of life. So if they're more along the bottom, then they kind of flatten out. If they're more predatory, then we'll see them more spread out to allow more amplification of the sound. Okay, so that's your little tidbit, your little introduction to sound production in fishes. I am going to take you to a major part of my research, and probably one of the favorite fishes of people are pipefishes and seahorses. These are all members of the fish family Syngnathidae. So these are very popular aquarium fishes. It includes everything from sea dragons to pygmy seahorses to what you think of as a normal seahorses. And then these are called pipe fish. So like a pipe that you run water through. They are very similar to a seahorse, except instead of being bent, they're straight. And what is super cool about all these fishes is that the dad has a pouch on his belly that provides parental care. So it is the dad in this family that has a specialized structure that can provide protection to the eggs and developing embryos. It can regulate salinity. It can provide aeration. And some of the studies we did, it shows that it actually is like a placenta and that nutrition is passed from dad to the developing babies through this brood pouch. It's called a brood pouch. Now, in this family of fishes, in the family Syngnathidae, there are different types of brood pouch enclosures. So we have some of the fishes that just have the eggs glued to the bottom of dad. Then we have some of them, like the fishes I studied, that have 2 flaps that kind of come together in the middle. So they're making like a pouch flap. And then you have the seahorses, which have this completely enclosed sac, and they just have one little hole at the top. And so we believed that as the pouch enclosure increased, so as we moved to more enclosure, that it became more complex and that dad gave more to the babies, had a bigger role in taking care of those babies. And so the name that Sean can't pronounce is actually called Chincoteague Bay. And Chincoteague Bay is located on the eastern shore, right over the Maryland-Virginia border. If you guys have ever heard of the saltwater cowboys or the ponies that are out there, there were books. You guys are a library. You should read these books. Misty of Chincoteague, all the Misty books from long ago. This is the location for that. It's a national seashore. It's Assateague and Chincoteague. It's gorgeous. If you've never been out there, it is one of the few beaches that still, since it is a national seashore, it's still gorgeously protected, and so amazing to go around. And there is a center there. It has changed its name. Now it's called the Chincoteague Bay Field Station. And we worked out of that field station, and they have programs. If anyone's interested for their families or their children, they have programs that you can spend a couple days out there exploring. You can spend a week out there. You can send your children to camp. They have grandparent and grandchild camps. It is fantastic location, except the mosquitoes are terrible. But they have two species of pipefishes out there. They have what's called the northern pipefish, which is called Signathus fuscus. And they have the dusky pipefish, which is called Signathus floridae. So they had two different species of pipefish. Coloration, you can tell the difference. The real difference between the two is the northern pipefish had a shorter snout and the dusky pipefish had a longer snout. And so we were able to go out there and collect these fishes and look at what the function of the brood pouch was. And I could even get them to mate in my lab. So this is a video that I made of pipefish mating. So I'm going to let you guys watch them mate. So all those fishes together, there were 3 females and 1 male. The females have the wider girth at the top part because that's where they have all their eggs. And then the male, you could see the 2 ridges along the side at the bottom, and that was his pouch. And so it was that quick that the female was able to transfer her eggs into that pouch. And then he does that little shimmy shake for a while to move the eggs down the pouch, and then they could go again. He can actually, pipefishes are not monogamous, so he could get eggs from several different females in that pouch at one time if he wanted to. And so what is really cool is that we were the first ones who actually were able to look at differences in the pouch structure and to see how the pouches were different between these 2 species. So Fuscus, the northern pipefish, it has 2 pouch flaps, but each flap folds over and kind of makes 2 tubes. So these are very distinct from those. And if you look here, the eggs are kind of shaded because each individual egg is encased in its own membrane. I don't know if you can tell, but these are much more cloudy. These are a little bit more clear because each egg has a membrane over it. And you can even see the blood vessels from dad running right along the babies. So it's really high. Dad is doing a lot of work. This is very close to being a placenta, which is extremely cool for a fish. This guy over here, the two pouch flaps came together and just made one big enclosed chamber. And we actually would see yolk from eggs that died during development floating around inside the pouch. And so kind of the eggs that didn't make it would provide extra nutrients. We still saw vasculature though from dad. So it was really cool. And overall, when we looked at, so I'm gonna give you some data this time, but it's simple data. I'll take you through it real quick, and you don't have to know all the statistics. But we basically looked at fishes, male fishes that had eggs or embryos versus male fishes that were not pregnant, didn't have any. And we could see that lipids, proteins, and carbohydrates in that pouch were always elevated when they had babies. So they are providing nutrients somehow into that pouch when they have babies. And it's always, even when they're not with babies, they're still keeping a level higher than seawater. So they're always regulating what is inside that pouch, which is a lot of work. So we actually did this thing where we took latex and we injected it into the fish, to the male fish, and we were able to get casts of the brood pouch. And so this is a scanning electron microscopy. And you can see that this is one single egg inside of the northern pipefish. And the entire thing is encased by vasculature. So it is literally an egg where dad is making complete blood vessels around that egg to give it full nutrients, full protection. The other one, we saw it more like a cup structure. So it didn't go all the way around, but it formed like a cup where the egg or the embryo could sit inside and get some nutrients from it. So it's still pretty sophisticated there. And so these are showing you the stages of development of the babies. So they do start off with really large yolk sacs. And as they start to use up their yolk sacs, they start to look like little baby fishes. And so we injected radioactive isotopes of a protein into dad. And if we saw that radioactive isotope in the babies, then we knew that dad was transferring nutrients to the baby. That was the way, because there's no other way that those babies could get these radioactive nutrients into them unless they came directly from dad. So first we looked and we said, well, yeah, their nutrient concentration's going down as they get older. They're using up their nutrients to grow. And when we did the tracing, this was the amount of that protein, that labeled protein, that we saw in the babies as they grew. So they were taking more and more from dad as they got bigger and bigger. So it was definitely coming from dad. Dad does have a placental-like function, which is the first we've ever seen for a male in a species, let alone a fish. So they're really good dads, is what we're telling you. Now, my heart lives with sound production in fish. I think that's just one of the coolest things, that fish can communicate. They produce all these different sounds. And so I had to look at sound production in my pipefish as well. And some of the things I was interested in is all the problems that are going on in the Chesapeake Bay. So Chincoteague Bay is located very closely to the Chesapeake Bay. And the eelgrass beds where the pipefish live are threatened. They get a report card score from the Chesapeake Bay Foundation of an F. And there's a lot of chemicals in the bay, which we know about from studying that pouch function. But the bay also has a lot of issues with dissolved oxygen. So the oxygen levels in the bay are really low. And it depends on the season and what's going on to look at how the oxygen levels in the bay change. So if it's a nice drought year, we don't have a lot of the pesticides and herbicides from all the farm out on the eastern shore washing into the Chesapeake Bay. So the oxygen levels stay pretty high. So red is dead, by the way, in these figures. So red means there's no oxygen in the bay. Organisms aren't going to survive. In a drought year, we have little red. When it's very wet and a lot of chemicals wash into the Chesapeake Bay, the red increases. This was the current last summer, where you can see that about 40% of the bay is red or dead in August. So oxygen levels go way down in the Chesapeake Bay in the summertime. And so we looked at sound production of pipefish. You got another video. Now, again, I'm not a cinematographer, so you have to bear with me with my videos. But now you can watch a fish eating and listen to the sound production. So I'm going to let you check this out. So that's a short video. So they're eating brine shrimp. Those are just giant sea monkeys, if you ever grew sea monkeys. Those are big sea monkeys they're eating there. But if you noticed it, I'll play the different sounds. So this is showing you what the sound looks like. You can see the frequency and amplitude if you're a sound person or physicist. So this is the sound for Floridae, which was the dusky pipefish. And this is the sound for the northern pipefish. So they sound pretty similar. It sounds like knocking, right? And it's actually a bone in the back of their head, because you saw them kind of jerk their head back when they sucked in that brine shrimp. So it's a bone back there that rubs against each other to make the sound. And so I wanted to know, if we were to drop a hydrophone into the Chesapeake Bay, could we say that listening to clicks was an indicator of feeding activity? And so I looked to see, is there a correlation between how many clicks we hear and how many times they take in food? And from that video, I think you could see it was a very strong correlation. Almost every single time they take in a piece of food, they are making a sound. So it's 97% of the time for Floridae and 85% of the time for the Fuscus one. So it's a really great indicator. And it's also a great indicator for how much food they have consumed. So if you hear more clicks, then they have consumed more food. And this is wet weight. So we actually measured how much weight of the food they had consumed. So then we took the oxygen down, and we didn't take it down to 0 because we would kill them. We took it down. So normal is 5 milligrams per liter. We took it down to 2 and down to 1. And what you can see is that sound production and food intake decreased very similarly, and it decreased very quickly. So there is a potential for us to monitor sounds within the bay. And if sounds have decreased, we know that there is a disturbance, that there is a problem going on. We've even seen like choking behavior in these fish. We've seen them go up to the surface and try to get in oxygen as well. A lot of the military research, a lot of my friends who did sound production on fishes and stuff in graduate school, they've gone on to work for the military. And the military does monitor a lot of aquatic sounds. So they use it, again, to determine the health of an ecosystem. They use it just for, to determine if there's enemies nearby, if things get quiet and so forth. But a lot of the work is now starting to see if it's a non-invasive, an easy way to say, hey, is this a healthy ecosystem or not? Let's see how noisy it is. And so that's where we were kind of going with that. This is my husband and my friend out there when we were younger collecting pipefish. So this is how you would collect a pipefish. Eelgrass beds are very shallow. You just take a net and just go through and see what you got. And they're pretty. I don't know if you guys remember from last week, but seahorses don't swim very fast. So these guys don't really go anywhere. They're really easy to collect with a net. So now I'm going to change gears. I'm kind of letting the headlines take you away a little bit. So I've done research on the cichlids. They were gorgeous and had bright colors. Pipefishes and seahorses are fantastic. They're beautiful animals. I cannot tell you how many seahorse and pipefish, particularly seahorse stuffed animals and figurines and all kinds of tchotchkes people would buy for me. And then I had to explain to them that to do my research, I had to rip the babies out of the pouch and like collect blood from dad. So it kind of lost their fancy beautifulness of my research with them. So then I dealt with problems that were local to our area that were terrible and people were extremely concerned about. So since about 2002, there have been significant fish kills in the Potomac, Shenandoah, and local rivers every single year. And so first it was just massive fish kills. Like 80% of the smallmouth bass in the Potomac River were dying every April. And once we started to dig into this, we saw that the male smallmouth bass in this area they had eggs. And so they called this intersex fish. So these fish were males that were producing eggs and they were dying. And so we decided to do some research to try to figure out what was causing this issue. So we hung up in West Virginia and the edge over in Virginia as well. And we looked at some sites. So we had our main stem site was our control. And then we had a site that had a fish kill in 2004. And so that was like our historic one. And then we had one that had a fish kill in 2005. And we did this research in 2007, 2008. So this is like 20 years old now. And we've come, and the rivers have gotten better. And I'll talk about that. And so what we were trying to do was, that's what we wanted. That's what we looked at. And then we went to collect fish in 2006 and 2007, and all the sites had fish kills. So then we had no real good control, but at least we had historic data. So this problem has spread significantly. Every time we come up with a new control site that hasn't had a fish kill, the next year there's a big fish kill in it. So it's really difficult to work on these problems because they're spreading faster than we can collect the data. But let me show you what these fish look like. So this is what the fish look like when we catch them and they're dead. So here's a sunfish and a smallmouth bass. They have these horrible lesions on them. And this here is a male's testy. So all these little black dots are sperm. These are big, large areas. Those are the eggs. And so in case you didn't know, males aren't supposed to produce eggs in their testes. That's not the way things are supposed to go. So when we opened these up and we saw the size and the number of eggs in the male testes, it really freaked us out. So you can see the percent with lesions and where the lesions were on these fish. You can see how many were intersex males. I mean, the main stem had all of the males were intersex. It's very dominant, at least half of them. So what we did is it looks like dots. It is kind of dots. But what we did is we took, it's called the interior kidney part of the fish. And that's where the immune cells are produced in this fish. And so we want to know why they're dying. So what was causing suppression of the immune system? And so what we did was we did these are big gels, and it just separates out proteins. So every single dot is a different protein. And you can see this one is missing a bunch of proteins right here, where these have it. And then some of these you can see have bigger dots than others. And so what we did was we investigated what proteins were missing, what proteins they had more of, and what we were able to find out. So you can look here and see that these are the same proteins in each and how the amount of them varied significantly. And what we were able to find out was that some of the fish had stress proteins. And then some of the other fish had immune responses and other ones didn't. And so we were able to take this data and go back, and we got slammed and in trouble for it. But it looked like the high amount of poultry farms in this area were part of the contributing factor. Now, we were never able to pinpoint it at the time, but studies since then have looked at some of the different pesticides they use and some of the different other treatments they use on the poultry farms to see that when the spring rains come, what is getting washed in, and is it mimicking what we see? Most likely it is not one single chemical. It is a mixture of chemicals. But since we have done this study and drawn light to it, we have seen great improvement. And I don't think in the last 10 years anyone has necessarily ever seen the massive amount of fish kills that we saw before. So that is a significant improvement, just to have that knowledge, even if we don't have a solution. And I have shared with you guys so much today, and I didn't want to overwhelm you, but I wanted to give you a resource and tell you some of the fun things that I have started to do. This is my husband over here, this little guy smiling at me with the other fish shirt on. I, about 6 years ago, started this program called Trout in the Classroom. And it's all due to this lady. And this was my kid a long time ago. He's 19 now, so he doesn't look like that anymore. So this lady was my son's teacher when he was in 4th and 5th grade. He had the same teacher. They did this thing called a loop. And she came to me and said, hey, I really want to do this project called Trout in the Classroom, where we're going to grow trout all year. But I kill fish. And I know you like fish, and you don't kill fish unless you want to kill fish. So I need you to help me. So that was in the spring of 2019, and I wrote a grant, and we got it. And so in the fall of 2019, we started Trout in the Classroom program. And so around Labor Day every year, I go down to Elkins, and I go to the Bowdoin Fish Hatchery, and they give me about 100 trout eggs per classroom. And these are rainbow trout eggs. And I bring the eggs back to my schools, and we put the eggs in public school classrooms, and they hatch and they grow all year round. Now, these are not public school kids. These are my students at West Virginia University. So I teach a introductory general biology class, Bio 101. And in order to make this program a success, I need help. I need lots of help. So I have they're mostly freshmen in this class. I have them come to these schools, and they help set up the tanks. They teach the students about water quality. We go out to the field. I'll show you some pictures of us out at local waterways. And it's a great way to get these freshmen that are away from home involved in their new community and doing something that's fun, and just to get that connection. And it's also fantastic for the public school students to see that they can one day go to college and do cool things like this as well. And so here's some of my students, some of my classrooms. You can see my eggs are hatching. And the curriculum is everything. So we do what's called a STEAM approach. So we do science and math and engineering, but we also have incorporated a bunch of art into the program as well. And so you can see here, we taught students how to mix colors. So we've done an art collaborative where we only gave them the primary colors, and they had to mix the colors and make paintings of the life cycle of the trout. And then these pictures actually have been posted up in the laboratory space at the university to highlight this program. And our program here is the only one in the country where college students collaborate with the public school students. And the national network highlighted that because that was such a unique experience. They didn't have that. This was Halloween. She didn't usually dress like Mike Wazowski. So I took her on Halloween to the school. In any case, so we have really received notoriety about that. And I've even been able to get Trout Unlimited to come and participate. So they come to the schools and teach the students how to fly fish. They show them how to make flies and stuff. So we're trying to get recreation and physical activity into it as well, especially since right now they're stocking all the waterways in the state, right? It's the big trout stocking time. And so it is a nice way to get you out with recreation, especially in our gorgeous state. Now, it just so happened that our very first year of this program was the year COVID hit. So you can see here, this is March 19th. That's when COVID kind of shut down the world. And we had all these, it was our first year. I had 4 trout tanks in 2 different schools. And so we had to release the trout by ourselves. It was me and my boys. We went out, found a little creek or a little pond and released them, and we recorded it, and we had over 1,300 views. So it was very well received that first year. This is obviously our COVID years. So through the COVID years, we kept trout in the classroom going. We never stopped it. Even when schools were totally virtual and parents were picking up supplies, we brought the trout eggs out to the parking lot and had students checking them out. We still kept doing our art projects. We did some paper mache and so forth. We had students working the whole time. Taking you to today, I have 26 tanks across 5 counties in north central West Virginia. And even though you guys are outside my range, I don't know if there's any trout. I know the Eastern Panhandle contacts me all the time because there's several programs out there. But I am on the nationwide Trout in the Classroom group. We have meetings a couple times a year. And I think if anyone in the state's ever interested in joining Trout in the Classroom, they usually get my name and I point them to resources or help out however I can. We have from first graders through youth crisis centers, middle schools, high schools. We have them all involved in the program, doing all kinds of activities. This is an ugly trout holiday sweater they made. We have releases every year, and our releases involve everything from having avian conservation come. We had Danielle Walker come when she was a House representative, and she had a reading station for us. We sample, we try on equipment. They, again, like I said, they do art. They do Valentine's Day projects to them. We have set up scarecrows. We have partnerships. So the DEP, Department of Environmental Protection, comes and assists us with some of our programming as well. And so I really have built this into a huge, amazing program that is involving a lot of individuals. Recently, we've teamed up with the Art Museum at WVU, and they help us do screen prints. So the students design their own trout, and then each classroom gets all their trout put together onto a screen print. And at our release, we have screen printing stations, and the students can either have t-shirts or posters, or the teachers get bags with their trout screen prints on them. So that has become a really big, fun station that we have come up with. We've done a quilt project for Mountaineer Week. So we've had students create their own quilts for a classroom. This happened to be the 100th day of school, so all these old people are first graders that are 100 years old. But they made their trout quilt squares, and we had them again displayed for Mountaineer Week. We also had them in the Morgantown Arts Center as well. And every year we try to choose a different theme for our quilts. So one so we're always it's a very exciting project. And again, these are my college students that I've taken out to the field. They have taught these elementary students how to use nets and so forth. And the thing about my college students is that none of them are biology students. They're like musical theater and business students. And because this is a general biology class, so that's not their field. But they love to get out there and have the hands-on opportunity and to have ownership in this project. I just kind of say, okay, this is how you use a net. Now teach them how to use it. And so it's been a lot of fun. If you're interested in the project, we have a Facebook page. It's called North Central West Virginia Trout in the Classroom. It's a private group. So you'll have to ask me, if you want to join, you just ask for requests, and me and a teacher at Bridgeport High School run the page. And so we post in there curriculum to do and how the fish are doing and so forth. So it's kind of a connection between all of us. And this is me throwing some teachers out in the water as well. And so if you have any questions about anything I have done, I left about 10 minutes for questions. This is my email at the university. Feel free to reach out if you know any teachers that might be interested in trout in the classroom. Again, I'm happy to point them in the right direction. It is an amazing awarding project. If you have any questions about sound production of fishes or about opportunities at the Chincoteague Bay Field Station, fish are fantastic. So I am always happy to share any resources that I can with you guys as well. And I'm going to stop sharing there so I can see you and then see if you guys have any questions for me.
If you do have questions, just raise your hand and I will come to you with the mic. I know we have one right here.
Hi. Last week you talked about the age of fish, and then this week you said something about the rings on their ear bones. How do you know when a fish is 500 years old or 20 years old?
So yeah, it would, to age a fish the most definitively, it would be to look at the ear bones, to look at the otoliths. So that's how they usually age them. But again, if you're going to age it that way, the fish is dead, right? You got to get it out of its head. And so they would look and count the rings on the otoliths. I mean, the only fish that's going to be 500 years old is that Greenland shark. Most other fish, I mean, for general purposes, most fish you think of in the wild are a max of 2 to 3 years. They just don't have a long life cycle. That's why fish have so many babies. They have a ton of babies because they're hoping that 2 live. Like, that's the hardest thing with trout in the classroom. I give these teachers 100 eggs, and they expect to have 100 fish to release. And I try to tell them, if you have 2, consider it a success, because fish don't have 500 eggs so that they have 500 babies. They have 500 eggs so that they have 5 babies. That's the trade-off. We have 1 egg released a month because we want 1 baby. So it's just, that's the trade-off in the way that organisms reproduce.
Any other questions?
The intersex with the fish, has that caused a decrease in population since the males are dying because they're being filled with the eggs and not reproducing with the females? So it's actually, if anything, it's decreased reproduction because the males,
their testes don't work well. So those eggs that they produce are not fertilizable. They're not good eggs. They're bad eggs. And then since they have them in there, it impedes their testy function. One thing I didn't tell you guys about in that area, so up in that area where those rivers are, there's a higher than national average rate of cancers, of like estrogen sensitive cancers. So people in those areas have higher rates of like brain cancer, breast cancer, prostate cancer than the national average. So when this first started happening and we started to investigate it, they were really centered on this, not necessarily because of the people in West Virginia, but the Potomac River runs through Washington, D.C. And there's a lot of important guys in Washington, D.C. that don't want to drink the water where the male fish are all of a sudden growing eggs. And so that got them to clean things up and start to investigate this really quickly. It's interesting, though, you know, all your wastewater treatment plants are supposed to clean up your water, but they just clean it up from harmful bacteria. They don't look at any of the chemicals, the pharmaceuticals, any of that stuff that's in there. They don't even measure it. Not to scare you guys.
Very scary. Any other
Yeah.
questions? No questions? All right. Well, thank you so much for being here and presenting. Thank you, everybody, for coming. Have a wonderful evening. Thank you.
Thank you.
Next week's class will be sharks.